Okay, hello everyone. Am I audible?
Yes, 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 sir.
Yeah. Okay, so today we have very less people. We have only six people here. So now it is already 3:10. So I think we can start with our discussion. So basically, like all other weeks, the discussion will be divided into two parts. First part will be the assignment, and the second part will be the discussion about some selected topics. And of course, all of you are always welcome to ask any doubt if you have regarding this week's uh, discussion or the this week's lecture content. If you have any, if you need any kind of clarification, you can ask always. Okay, so uh, so now we can start with our session. So our session will be based on the week 6 of the NPTEL lecture. So that actually discussed about the SMC complexes, the chromatin organization. Yeah, so regarding this, regarding the role of different SMC complexes in the chromatin structure organization. So we will discuss about that. Some selected topics will be covered, and also we will discuss some previous year's assignment question. Uh, so let's start with this. Okay. So can you see my PPT? Is the PPT is visible and full screen? Can you can you please confirm?
Yes, yes, sir.
Okay. Okay. So yeah, fine. So let's start with our discussion. So here I will discuss about some assignment question from previous year. So and I mean the uh, I will discuss about some assignment question from the week 6 previous year NPTEL lecture. So in this list, the first question is which of the following protein prevents the expansion of the position loops of cohesin, of course. So the options are ECO1 protein, WPL1 protein, MCD1 protein, and SMC1 protein. So which one should be the right option? For cohesin, which protein is responsible for preventing the expansion of the positioned loops? What will happen if the expansion of the expansion of the loop will be happened in cohesin? What will be happen? Cohesin will be in the chromosome, or it will be uh, deloaded from the chromosome. What do you think? Can anyone please answer for that? Any idea about that? Anyone? Any answer? You want to try? So, uh, if the expansion of the loop position loop will be happen in the cohesin, then of course the cohesin will not be able to be present in the chromosome, right? So it will be deloaded. So that's why if you want to stay your cohesin in the chromosome, then you need to prevent this, right? Is anyone there? Hello.
Hello, sir. Sir, I'm a little bit behind on the schedule, so I'm sorry.
Sorry.
I'm a little bit behind on the lecture, so not. I don't know this.
Okay, okay, no problem, no problem. Here you can just guess. Okay, it's of course fine. Okay, no problem. Okay, so let's come to the first question, that which of the following protein prevents the expansion of the position loop in cohesin? So if there is why this prevention if there is expansion of position loop of cohesin, means the cohesin can entrap the chromosome, right? Cohesin can entrap the chromosome. So if we want to deload the cohesin from the chromosome, then we need to prevent this expansion of the position loops. For that, WPL1, there are many options: ECO1, WPL1, MCT1, SMC1. So WPL1 is the responsible for deloading the cohesin complex from the chromosome. Okay, so I will just discuss about here what actually WPL1 does. Okay, so from this, yeah, so from this slide you can see that here, for maintaining the cohesion, for maintaining the entrapment of the chromosome inside the loop of this cohesin, you need ECO1, because ECO1 will acetylate this SMC1, which will maintain the cohesin ring close. But here WPL1 what will do, it will opens the SMC3 SMC1 complex. This is SCC1, this is SMC3. So it will open this SMC3 SCC1 interface, and as a result of this, the cohesin will be now open complex, and it will be deloaded from the DNA. So that's why for this, if we discuss about this question, the right answer will be WPL1, because WPL1 is responsible for preventing the expansion of the position loop, and because of that, cohesin can be deloaded from the chromosome. Okay. Okay, so the second question is, why was ATP gamma S used in the control experiment in the double tethered DNA assay with the condensate molecule? Okay, so I hope you have gone through the lectures, and there was one experiment where they did the double tethering of the DNA in a plate, and they found the and they tested the loop extrusion activity of the condensate and ATP. So if you went through the lecture, so you must have known. Okay. So the question is, why was ATP gamma S, which is a derivative of ATP, was used instead of ATP in the control experiment of the double tethered DNA assay with the condensate molecule? So here basically they wanted to check the loop extrusion activity of the condensate molecule, and the target of the condensate molecule was the double tethered DNA, and they probed for the loop extrusion activity of the condensate molecule. For that, they use the ATP gamma S, which is a non-hydrolyzable ATP. Okay, but the normal ATP is hydrolyzable ATP, but here they use is the non-hydrolyzable ATP. So can anyone tell me that why this non-hydrolyzable ATP was used instead of the normal ATP? What would be the problem if they use the normal ATP? Suppose in any experiment, some like here condensing requires ATP to mediate its loop extrusion activity, right? So it requires ATP. Actually, it requires the ATP hydrolysis, right? So here, for this purpose, when they did it in vitro, they use ATP non-hydrolyzable analog. Why? Why is the question? The why they have used this non-hydrolyzable ATP? Why not normal ATP? Any idea? If you have any idea, you can just tell me. Any idea? Just try. Anyone want to try? Can you guess anything from these options? It is easily hydrolyzed by the condensate molecules into ATP and S. Any idea from this option? So the answer is the reason of using this non-hydrolyzable ATP is that it can bind, condensate can bind, I mean ATP, that ATP can bind the ATP binding region of the condensate, but it will not get hydrolyzed. That's why it is non-hydrolyzable analog of the ATP, so it will not be hydrolyzed. So if in your experiment, like if your protein condensing, like here, if your protein requires ATP hydrolysis, then your ATP should be in the, or if your molecule, I mean if your protein requires ATP as a cofactor, so for that ATP should be in the form of ATP. But a normal ATP will be easily hydrolyzed. So after hydrolyzation, ATP will be converted into ATP and PI, right? So there will be no more ATP after some time, because ATP hydrolysis will be occurred and it is a spontaneous reaction. So it will be occurred readily, right, spontaneously. So for that, the protein, your protein, like here condensate, it will not get ATP, for a very long time, so that you can do your assay. So if you are doing an assay by using a protein condensate and its cofactor ATP, then if you want to catch the time frame for some particular time, so for that reason your ATP should be in the ATP form, right? So for that reason they have used non-hydrolyzable analog of the ATP, so that ATP will be in form of ATP. ATP will not be hydrolyzed, and it will not be hydrolyzed to ADP and ATP, because if it will be hydrolyzed, then it will be converted into ADP, so there will be no more ATP. That's why they have used the non-hydrolyzable analog of the ATP. So the option is it can bind the ATP binding region of the condensate complex but does not get hydrolyzed. That's why they have used ATP gamma S. Is it clear, or any doubt if you have? Any doubt you have, please let me know. Is it clear what is ATP gamma S? Okay, can anyone of you explain that why this non-hydrolyzable ATP was used instead of normal ATP? Anyone want to explain?
Like, so that we can identify the ATP. Like normally, like ATP will get hydrolyzed to
Yes.
ADP. So, but in
Yes.
this case, the ATP will remain as ATP.
Yes, so that the ATP will be in the form of ATP, right? Not in the form of ADP. Okay, fine. So let's move on to the next question. Pick the correct statement about the SMC complexes. First option is they are essential chromosomal associated protein complex present only in eukaryote. Is it true completely? Is it true?
They're conserved, right? So, present.
Yeah. So they the first statement is telling that they are present only in eukaryote. So it is not true, because in prokaryote it is also present, right? So that's why it is conserved. So in the second option they are telling that they are conserved. Okay, fine, non-essential. This is true or false for the SMC complex? Are
No,
they?
it's essential. It's very essential.
Yeah, it is essential, right? So that's why this statement is also false. Third statement, conserved. Okay, we found that conserved, essential, yes, and chromosome associated protein complex. So it is the right option, right? Because conserved, essential, and chromosome associated, they are known to be recruited to the chromosome. So it is the right option. Fourth option is conserved, essential, but membrane associated complex, but it is not true because it is chromosome associated protein complex. So the answer is the option C. Okay. So next question. Again we have four statement. We need to find the correct one for the SMC protein domain organization. So which one will be the true? If you remember the SMC protein architecture, which option is the true option? Which one is the true option? Okay, so it has N-terminal Wakare motif, it is true. Coil region true. Hinge region true. Coil region again true. And C-terminal Wakar B motif. So it is a true, right? But in the option B, N-terminal Wakar B, that is false because it has N-terminal Wakar A and C-terminal Wakar B. It is false. Third option is, in third option everything is fine, but there is only one coil region. So it is not also true because it has two coil region. Okay. Option D, N-terminal Wakar A motif, hinge coil region, C-terminal Wakar B motifs. Again there is only one coil region, so this option is also not true. The true option is the first option, Wakar N-terminal Wakar A, coil hinge, again a coil and C-terminal Wakar B motif. Any question here? Any doubt? For your more clarification, you can see this is the basic structure of SMC. There is a N-terminal Wakar A, C-terminal Wakar B, hinge and coil region, two coil regions. So that's why the first option is the correct. Okay. Any doubt? Okay, so the next question is the chromosome conformation capture technique can be used to visualize the genomic organization at the high resolution, detect spatial relationship between known DNA sequence, and to yield information rich data describing genome topology, and all of the above. Which should be the right answer for that? Any idea? Anyone want to? Okay, so let's go by option. Okay, first is, what is the application of this chromosome conformation capture technique? First is to visualize genomic organization at high resolution. It is true because at high resolution it can visualize the genomic genomic organization matlab, I mean here the by using this technique you can say that where a loop is formed, where some other secondary structure has been formed in your entire chromosome. So that's why you can visualize the genomic organization at the high resolution. And then the same thing is the option 2, that spatial relationship, because in a spatial relationship means the 3D structural pose of the structural organization of your chromosome. In some place there is loop formation will be occurred. So so that's how you can find the spatial relationship between the known DNA sequences. And the third option is also true because to yield information rich data describing genome topology. So it can describe the genome topology. So that's why the all of these three options are right. So that all of the above will be the proper answer. Okay. Next question. The cut mutants in fission yeast have the following phenotype: bisected nucleus during cytokinesis. Is it true? Bisected or fragmented nucleus, broken nucleus, incomplete length nucleus during cytokinesis. Is it true? What are the cut mutants? Cut mutant are the mutant in the yeast. In this mutant, they have a phenotype of fragmented DNA or the bisected nucleus. Okay, so their DNA will be fragmented. That's why they are this due to this phenotype, the mutants are known as cut mutants. So, which of the phenotype can be the phenotype of this cut mutant? Bisected nucleus during cytokinesis, or the bisected nucleus during interphase, or during G2? Any any idea? Okay, so the answer is bisected nucleus during cytokinesis. Why cytokinesis? Because after cytokinesis, uh, the nucleus will be bisected, right? Because there is a possibility that the anaphase breach that may not be resolved, or yeah, the anaphase breach that cannot be resolved. So due to that, it will have a double. If the cytokinesis will be occurred, then it will have a immature double strand break in the DNA of the progeny. So that's why they have the bisected nucleus during cytokinesis. This is the phenotype. Okay. The next question is, which of the following are true regarding SMC complexes? They have only homodimeric structure. Is it true? Homodimeric structure. Can anyone please try? Okay, so so SMC complexes does not cannot have the homodynamic structure, right? Because homodynamic means there is similar to same two subunits will come and form a complex. So this is not the case for the SMCC complexes because they are all first of all they are not dimeric. There are more than two number of proteins involved. And the second thing is they are not homodimeric or because all the participant of the complexes all the proteins which form the from the uh, SMC complexes the all of the proteins which are known so far they are not homo, all of them are different. That's why they are not homo and they are not having the dimeric structure. There are more than two number of proteins involved, so that is not the true option. Secondly, structure of SMC complex is X or X shaped or cross shaped. Is it true? It's not true, right? Because the SMC complex has a structure of ring type structure, right? It is a ring shaped complex. So this is not a X shaped complex. That's why this option is not true. Third option is it has ATP binding domain which catalyzes ATP hydrolysis. Is it true? Is it true? Just remember the structural organization of the SMC complexes. Or if you can, if you want to see, you can see this slide too. See, SMC complexes, Walker A domain, Walker B domain, coil hinge, and you know that the folding after the folding along the hinge, after the folding this will come here. So these two loop region will form a coil region, and Walker A and Walker B region will be come to the close proximity, and they will make a functional ATPase domain. Right, this is the basic folding process of the SMC complexes. So now after that, try to answer this question that that SMC complex about the third option, SMC complexes have the ATP binding domain and which can catalyze ATP hydrolysis activity. It is right, because it has the when after folding they have a functional ATPase head domain. So ATPase domain, as we know that it has a ATP binding region which and it can catalyze the ATP hydrolysis reaction. So for that SMC complex should have, as they has the ATPase domain, so it should have the ATP binding domain which catalyzes the ATP hydrolysis. So this option is the right one, third option. Any doubt? Please ask me. Okay, I think we missed one question here. Yeah. The condensate complex participate in segregation and histochromatic decatenation. Is it true? Is it true? Option A. Okay, today I'm not getting so many responses unlike the other days. Any problem here? Can anyone speak up? Because today I am getting very low response from you, unlike the other days. So, is there any problem or issue?
So we have not joined previous lectures.
Sorry, you have not?
Joined previous lectures.
No, I mean, did you watch the lecture content of this week 6?
No, I haven't watched.
Okay, so that's why I think you are facing. So, how many of you are there who has not watched the week 6 lecture content? Can you please let me know? How many are there? Okay. Okay, so I know it is tough to uh, cope up, because if you have not watched, then it is quite difficult for you. Okay, so just try to just follow me, and please ask me whenever you have problem. Don't just let your doubt inside you. Just ask me if in any place you have any problem in understanding. And after this, we are remaining of C4 question. After this, we will again we will I will explain you some brief topic about this week 6 lecture content. Okay, I think after that it will be easier for you. Okay, so this question is the role of condensing complex participants in. They have given us many options. So the first option is segregation and histochromatic decatenation. So what is segregation? As you already know that in anaphase, the two sister chromatid will be segregated. They will be pulled by the microtubule, and they will be segregated to the, or they will be pulled to the opposite pole of the cell. Right? That is segregation. The second is histochromatic decatenation. Someone is there. Huh. So yeah, so another thing is this, the sister chromatid decatenation. Okay, decatenation means when after replication, when there is two sister chromatids are there, then they will be interlinked with each other by this orientation. Okay, so they will just be interlinked with each other. Okay, so if there, so that is called the catenation. So if this catenation will not be resolved, then they will not be properly segregated, right? So for that, this interlinking or this catenation should be resolved. So condensing complex was found to have a role to resolve this process, or which is also known as decatenation process, because then only the sister chromatid will be segregated, and that's why it has role in the sister chromatid segregation. Second is, which you already know, that it has a role in maintenance of the genome organization, the structure of the genome, the loop extrusion, the to make the chromosome more contains. So all of these things are these roles are played by the condensing complex, which can actually compact the DNA, to give that your chromosome a typical structural organization. It will compact the DNA, it will make your DNA contains, and it will help you to get a structurally organized chromosome. Third is clustering of tRNA gene. It is also true, because see just a minute. I think someone. I will continue just. Yeah, yeah. So the third option is clustering of tRNA gene. This is also true. Why? Because in especially if we consider about the budding yeast scenario or our scenario, there were several tRNA genes are there, but these tRNA genes are located in different chromosomes, right? So when the chromosomes will be segregated, then all of these tRNA genes will be segregated by uh, I mean, all of these tRNA gene will be segregated at a single point. Okay, so all of the tRNA gene will be come together. They will make a cluster, and this cluster will be segregated with together. So all of the tRNA genes will be segregated together. Did you understood that? What is clustering of tRNA genes? See, you have different tRNA genes in your chromosome, right? And it is also true that all of the tRNA genes are not located in a single chromosome. So all of the tRNA genes are actually scattered within different chromosomes. So when the chromosomes will be segregated, then all of the tRNA gene from different chromosome will come to a single point, and they will make a cluster, and after forming this single cluster, they will be segregated as a cluster, as a group. So that's why it is called that making cluster of the tRNA gene and segregation of this cluster of the tRNA gene. That is the wild type phenotype of segregation. In segregation, for proper segregation, this tRNA gene clustering is required so that all of the tRNA genes will be segregated at a single cluster. And condensin was found to have a role in the clustering of this tRNA gene, to maintain the cluster of the tRNA gene. Fourth option is, I think you know that condensation complex of course has a role in chromosomal condensation. So all of these options are true. So the answer is all of the above. That we have already discussed. So yeah. So the next question is, nucleosome consists of, I think, nucleosome consists of DNA, only histone, both DNA and histone, and none of the above. So what do you think? Nucleosome, do you remember the nucleosome structure, bead string model?
Both histones and DNA.
Both histone and DNA, right? So the option C is the right answer. Histone and DNA will be, and the histone will be wrapped by the DNA. So both the histone and the DNA are the option. Okay. So the last question is, okay, yeah. So we are finished with our question. So just let me know that if you have any doubt with the discussed question. If you have any doubt, just tell me. In any question. Any doubt?
Good afternoon, sir.
Yes, good afternoon.
Sir, once again, 7th and 8th question, sir. Explain, sir, please.
Sorry?
7th and 8th question.
7th and 8th question.
What's
Okay,
that?
okay. Yeah, fine. This one, right? Okay, so yeah. So she requested for the question 7. So the question 7 is asking that the cut mutant in Pombe have the following phenotype. So what is the cut mutant? In the cut mutant, the phenotype of this cut mutant is their chromosome will be fragmented, or they have the fragmented DNA. That means they will not have the complete length of the DNA. Okay, so their DNA will be fragmented, not a continuous. The integrity of the DNA will be lost. So what can be the phenotype of this cut mutant in the S. Pombe is a fissionist. So what the phenotype of this cut mutant in the Saccharomyces pombe? So the first option is bisected nucleus during cytokinesis, bisected nucleus during interphase, bisected nucleus during G2. So the correct option is the bisected nucleus during cytokinesis, because after cytokinesis you will see the bisected nucleus, because the or the fragment which will be formed, which is the phenotype of this cut mutant, they will be formed only after the cytokinesis. When there is when the the division, after the cell division, when the cytoplasm will be divided in the process of cytokinesis, then you will find the fragmented DNA, which is also known as the cut mutant phenotype. So that's why bisected nucleus during cytokinesis is the right answer. In the interphase, you can't see the bisected nucleus. In the G2, you can't say, until and unless you will facing the cytokinesis, then only you can see that the phenotype that of bisected nucleus. Okay, so this is the phenotype of the cut mutant in Pombe. Does it clear your doubt? Is it fine? Hello, am I audible? Hello.
Sir?
Yeah, so does it clear your doubt, or do you need any more explanation?
No, sir. Thank you, sir.
Fine, right? Okay. So the next question is what she asked, that which of the following statements are true regarding SMC complexes? Okay. So for that I will just show you this slide again, and then you will tell me. See, this is the SMC complex architecture. It has an N-terminal Wakare domain, C-terminal Wakare domain, and a hinge region, and there is two coil region. So when the SMC complex will be folded along the hinge region, then this will come here. So this and this region will make a coil region, and this C-terminal Wakare B and N-terminal Wakare domain will come to close proximity, and they will make a functional ATPS domain. So now after seeing this slide, now you will tell me that which can be the following, which can be the correct statement about the SMC complexes. Is it a homodimeric structure? Do you think? Is it a dimeric structure, SMC complex? No, right? Because it has more than two number of protein. Okay, so it cannot be dimeric. So this option is false. Second option is the SMC complex is cross shaped. It is also not true, because as I saw you the slide, the SMC complex structure is ring shaped, right? Ring shaped structure. So that is not the X shaped structure. The third option is SMC complexes have ATP binding domain which catalyzes it. This is true, because as I told you that after folding there is ATPase head domain. Okay, so in ATPase domain they have the ATP binding region and which catalyzes the ATP hydrolysis. So the option C is the right statement about the SMC complex. And the option 4, that it does not have any associated non-SMC complexes when bound to the chromosome. This is also not true, because because in SMC complexes there is not only the SMC proteins, there are non-SMC proteins are also there. Like in SMC 5-6 complex there are NSC1 and NSC3 which are non-SMC complex, or in cohesin also there is SCC1, there is SCC3, PDS5, so that also non-SMC subunit. So in SMC complexes there are additional non-SMC subunits in addition to the SMC proteins. So this option is also wrong. So the right option is the option third. Yeah. So now is it clear? Option 7 and the question 8 and 7, it is clear now?
Yes, sir. Thank you, sir.
Any more doubt? If you have anyone?
I think, sir, the answer is both histone and DNA molecule.
Yes. The answer is both histone and DNA molecule. Correct. Yeah. So any doubt you have till the questions we discussed? Any doubt or any problem in any question? You tell me. Okay, so I think you don't have any doubt for that. Okay, so let's move on to our next section where we will discuss about some selected topics from this week 6 lecture content. Okay. So if as I already came to know that many of you have not seen the videos, so if anyone have seen the videos, can so then if you have any doubt in any video, you can you can ask me. So is anyone there who have watched the video and have any doubt in any section? If you have, you can ask. Okay, so you don't have any doubt, right? Okay, so let's start in the second half of our session, which is uh, yeah. So we will start about the nucleosome organization. So as the week 6 lecture contents were about the organization of the of your chromosome, how it is being structurally organized, and the role of SMC complex in organizing this chromosome. So that was the main or the the topic of discussing the week 6 lectures. So for that, so initiate about that, we can first discuss about nucleosome organization. How the nucleosome. So nucleosome, as I told you, some of you actually told me that nucleosome is made up of histone and DNA, right? So here you can see that this is a popular bit string model of nucleosome, where this histone proteins blue color, they are actually playing the role of bead, and around the bead this DNA is wrapped like a ribbon. So that's why it is also known as the bead string model, where a string, if you will tie a string, if you will wrap a string in the bead, then the same thing is created here. The DNA will be wrapped around the histone, and this DNA plus histone, this structure is known as nucleosome, and that is the first level of compaction of the DNA. So that is called a nucleus. So this structural organization is known as nucleosome organization. And the histone. So now let's talk about something about this histone proteins or this histone subunit, which is acting as a role of bead here in the nucleosome. So histone in this bead, there is a histone octamer. So how this octamer is formed? Here first two subunit of the histone H3 and H4 will come and it will make a H3H4 dimer. Here H2A and H2B, these are two subunits of histone. They will come and they will make a dimer. And after this dimer formation, this H3H4 dimer and H2A H2B dimer, they will come and they will make a H3H4 trimer, where there will be one H3H4 tetramer. Okay, and when this H3H4 tetramer will come and bind with this H2A H2B dimer, then it will form a histone octamer. Yeah, so that is the formation. That is the how the histone octamer is being formed. That is depicted here. So first H3H4 will come to make a dimer. Then again this one H3H4 dimer and another H3H4 dimer will come and will make a H3H4 trimer. Similarly here H2B come, it will form H2B dimer. After that this H2A after that another H2B dimer will come to make a H2B tetramer. And after when this H2B tetramer will form and H3H4 tetramer will form, they will come and form this histone octamer when there will be a H3H4 tetramer and H2B tetramer. So total octamer. Okay. Okay, someone has raised your hand. Abhishek. Yeah, so what question do you have, Abhishek? You have raised your hand. Hello. Hello. Okay. Okay. So let's discuss one more time this histone octamer, how this histone octamer is formed. So first step, H3H4 will come, make a dimer. So now two kind of this H3H4 dimer will come and make this H3H4 tetramer. Here H2H2 will come and form a H2H2B dimer, and two H2H2B dimer will come and form the H2H2B tetramer, and this H2H2B tetramer and this H3H4 tetramer will come and form a histone octamer when there will be one H3H4 tetramer and another H2H2B tetramer. So total there will be a histone octamer. So any doubt in this histone octamer complex? You got it? Did you get it?
Yes, sir.
Okay, fine. Okay. So now let's discuss about some SENP-A. Okay, SENP-A is a analogous to the histone H3 complex. Okay, H3 protein. Here H3, as you know that H3 is a subunit of the histone octamer. So this SENPE, these are actually analogous to the H3, and this SENPE, it has a specific or a unique activity that it can be recruited to the centromere, and if this SENPE will be recruited in the centromere, it will play the role of H3. Like a H3, instead of H3, the SENPE will be recruited here. And why is it important in the centromere? Because in the centromere, you need SENPE here in place of H3, because this SENPE will actually uh, mediate the recruitment of the kinetochore complex. And as you know that for the segregation, you need the kinetochore complex recruitment to your centromere. So for that, SENPE is very much important. Without this, kinetochore cannot be recruited here. Uh, yeah. So, oh, okay, yeah. So as I told you that in centromere, instead of H3, there will be SENPE recruitment as a part of histone octamer, because because in centromere it will, yeah, because in centromere the role of this SENPE requirement is to recruit the kinetochore complex. So how is it working? So first of all, this SENPE is a part of histone fold domain, and this it has this region CATD, which is known as SENPE targeting domain. It is recognized by HJURP, which is a chaperone of this SENPE. Okay, and this recognition of CATD domain by this HJURP actually helps to recruit the SENPE in the centromere. So for recruiting of the SENPE in the centromere, you need the CATD domain in the SENPE. If you will mutate the CATD from here, your SENPE will not be recruited to the centromere. And if there is no centromere, and if there is no SENPE in the centromere, there is no kinetochore complex assembly and there is no proper segregation. Okay, so that's why this CATD domain is required, which will be recognized by this HURP, and that will actually lead to recruitment of the SENPE to the centromere. Okay, and SENPE, in addition to the CATD, it has two more important domains. One is C-terminal domain, CAC domain, which is known as SENPE C-terminus, which is highly enriched of hydrophobic amino acid residue that directs the kinetochore assembly by binding SENPC. How? When the SENPE will be recruited to the DNA by the virtue of this CATD domain, when the SENPE will be recruited to the centromere actually, then the CAC will bind to the SENPC. SENPC is another protein. So CAC domain of SENPA will bind to the SENPC, and the SENPC, it can make a direct contact with the kinetochore complex. So it will act as a landing platform for the kinetochore complexes. That's why the CAC domain of the SENPE is very required to recruit the kinetochore complex in the centromere. Another important domain is domain of SENPE is the N-terminal domain. So in N-terminal domain, it has been found that this N-terminal domain also can recruit the SENPC in the centromere, but it needs the interaction of the SENP. Only in the presence of SENP it can recruit the SENPC in the centromere, and the role is same to act as a landing platform for the kinetochore, so that kinetochore assembly can be occurred in the centromere. So that's why SENPE, by this mechanism, you can see that by this mechanism SENPE has a role to recruit the kinetochore complex in the centromere region, so that the microtubule can come and attached. So that is the role of SENPE as we discussed here, which is a H3, which is analogous to, which has a domain which is analogous to the H3. So any doubt in this explanation? Please tell me. Any doubt? Why CENP is required in the centromere and how it can help to, recruitment of the kinetochore complex. Is it clear? Is it clear? Any doubt? Please tell me. Is it, so is it clear to everyone? Okay, so I think it is clear to everyone. So I will just move forward. So now we will discuss about the SMC complex architecture. So here, if you will see this figure, here there is SMC complex structure where there is a N-terminal Walker array, then there is a loop region, then a hinge, then again a loop region, and then again a C-terminal Walker B region. So after the folding, so how the folding occurs, based on this hinge region, this region will come and come to here. So this two loop region will form a coil region like you see here, and this Walker A and this Walker B will come to the close proximity and form this one. And when this Walker A Walker B come to the close proximity, it will make a functional ATPase domain. So that is the major architecture of the SMC proteins and how they fold. Similarly, and it is same for the all of the SMC proteins, like is cohesive, there is SMC 1,3 folding pattern is same for condensate, which contains SMC 2 SMC 4, and same for SMC 5,6 complex, which contains SMC 5 and SMC 6. So all of these 6 SMC proteins are known to have the same kind of structure and their folding is same. So that is the basic architecture and folding of the SMC proteins. And so there are three cohesive proteins. So there are three SMC complexes will be discussed here. One is cohesin. Cohesin, there are two SMC proteins, SMC1, SMC3, and also there are some non-SMC proteins like SCC1, which is a glycine, which will make the ring close. It will interact with the SMC1 and SMC3 and make a close complex. And also there is PDS5, SCC3. In condensate there is SMC2 and SMC4, and they have BRN1. Like SCC1 is a glycine here, it will act as a gate of this. It will make the cohesin close complex. Like that, there is BRN1 in condensate. BRN1 will make contact with SMC2 and SMC4, and it will make this complex close complex. And also it has other two non-SMC molecules, YCS1 and YCG1. Both of them are known to contain the heat treatment motif, and these heat treatment motifs are actually it has the helix and helix motif. So as you know that HTH motif are DNA binding domain. So this YCS4 and YCG1, they help to bind the DNA. So that's why they can recruit the condensate molecule into the chromosome. And there is SMC 5,6 molecule. Here you can see that there is SMC 5, SMC 6, and also there is some non-SMC molecule as NSC1, NSC3, NSC4, and in the hinge region there is NS5 and NSC6, and this coil region there is NSC2, which has a enzymatic sumo ligase activity. So this is the SMC complex architecture, and three different SMC complexes are discussed here. So yeah, sorry, doubt regarding this? Any doubt? Is it clear to you? Can anyone want to explain that? You can take any of the SMC molecules, like you can take cohesin, and you can, if you want to discuss the structure of the cohesin, can anyone come and discuss? Anyone want to? Anyone please come and discuss? You got it right? Okay, fine. So yeah, so I think you understood it. Okay, so let's move on to the next slide. So here now, as we know that these SMC molecules are known to be recruited to the chromosome, they bind to the DNA and they do their work, right? So now it is, if you are working with these SMC complexes, so there is an important assay for finding the chromosomal binding of SMC molecules, and that assay known as CHIP, chromatin immunoprecipitation, where we will found where actually we will find that actually we can do the quantitative assay that how much binding of a particular SMC proteins are there in the chromosome. So what is the technique of this CHIP assay or the chromatin immunoprecipitation assay? So this is your DNA, and your DNA, as you know, that there are many proteins are known to bound to DNA. Okay, now your dynamic protein of interest is this one. Okay, but there are many other proteins also. So now your goal is to, yeah, so after that your goal is to, for a particular region, if you want to check the binding, or if you quantitatively measure the binding of your protein of interest, then what you will do? So first of all, you will do a in vivo cross-linking of the protein DNA complexes. That is done by the formaldehyde. So it will basically, what it will do, it will make a covalent bond between the DNA and your protein. So now all the proteins are covalently linked, because all of these interactions are the non-covalent, right? So the directions can be, they are temporary. So after some time, the proteins may not bind to the DNA, because it is non-covalent, temporary. So to make it permanent, we need to make it covalent, and that's why we did the cross-linking of the protein and DNA molecules. After that, we will sonicate the this DNA protein complex. Actually, we will sonicate the DNA to fragment the DNA. After the sonication, the fragmentation, we will see the this is the fragmented DNA. Okay. Now, after that, you can see that we did this one, right? This one. We did this actually we did this binding. Okay. Or if you want to know the, suppose for this protein of interest, you want to know the binding sequence of this protein, in which site this protein binds. So for that you can actually do this assay. So after sonication, you will do the immunoprecipitation by using antibody against this protein. After the immunoprecipitation, only this protein DNA complex will be immunoprecipitated, because antibody is specific to this one. So rest of this will be in supernatant, but this will be in the so only this will be precipitated. So when you centrifuge after that, all of these other protein DNA complexes will be in the soup, but this will be in the pellet, right? And actually what we do, we actually incubate with the bead, and the bead contain the protein A. Okay, protein A conjugated bead. So the protein A has a affinity to the antibody. So if we will incubate with the bead, then all of these two proteins will be bind to the bead, but all the other protein DNA complexes, they will not bind to the bead. Okay, only these two will bind to the bead. So after separating these two, we need to remove the first we need to wash to remove the non-specific binding, and then we need to remove the cross-linking by heating, and after that we will separate the DNA and protein, and we will find the DNA in which this protein was bound. So after that we can do a artificial analysis to find that how much binding of this particular protein in this sequence was there, or if you don't know the sequence of this binding site, we can do a sequencing, which is also known as ChIP-seq. So in the ChIP-seq we will do the sequencing, and we will find the sequence at which this protein of interest was bound. Okay, so any doubt in this process?
So sir, how is reverse cross-linking done? Like,
Reverse, yes, very good question. Reverse cross-linking is done by heating. We usually heat it at 65 degrees Celsius. Okay, and this, yeah, so in the 65 degrees Celsius heating, this covalent bond will be disappeared. It will be broken. So there will be no more covalent bond between the protein and DNA. So now the protein and DNA are separated. After that we will just isolate the DNA by using the PCI, phenyl chloroform isopropanol, which will see now. After that we only have the antibody, which is a protein, the bound protein, which is also a protein, and the DNA, but we need the DNA. So after decross-linking, when we will give heat to break this covalent bond, so after this decross-linking we treat this with a proteinase K treatment, so it will denature the protein and the antibody also. After that we do the phase separation by using PCI. So as you know that in phase separation, the denatured this protein will be, or the protease treated the proteins will be come into the phenol layer, and the DNA will come into the aqueous layer. So we take the aqueous layer which contains the DNA, and that DNA is this. After that we either do the RT-PCR to know the quantitatively measure of this DNA, and we can do the sequencing to know the sequence at which this protein was bound. Yeah, so that was after steps of the decross-linking. So is it clear now?
yeah, yes.
Okay, fine. Anyone has any doubt in this? ChIP, chromatin precipitation, how and why we do? Okay. Okay, so I thought, yeah. So now, after that, we can discuss a topic about cohesin loading and cohesion establishment. I think all of you know that about cohesin. Cohesin is known to be hold the sister or the entrap the sister chromatids within its ring, and when there is a onset of the anaphase, at this onset the this SCC1 subunit of the cohesin will be degraded, and the chromosomal segregation will be occurred. That is anaphase. So for that, for that, two important thing is that cohesin loading and cohesin establishment. Okay, so how this cohesin loading is occurred, right? Now cohesin loading is only found in the in early G1 phase you will not find your cohesin loaded into the chromosome. Okay, but in the late G1, when there is a requirement of S phase, when replication is just about to start, then you will find that cohesin will start to load into the chromosome, right? So how this binding is being controlled? So one hypothesis is that for loading of cohesin into the chromosome, the requirement is the total cohesin complex, means SMC1, SMC3, SCC1, SCC3, PDS5, which, and this total entire SMC cohesin complex bound to SCC2, SCC4. This thing can only be loaded into the chromosome. So now everything is fine for every but the SCC1, this SCC1 is only, this level or the expression of the SCC1 can only occur in the late G1. Okay, in early G1 you can't find SCC1. If the SCC1 production is not there in early G1. In late G1 it will start to produce. So as the cohesin loading requires the presence of this all of the cohesin subunits, so in the absence of SCC1 it cannot be recruited to the chromosome in the early G1. But in the late G1, when the SCC1 starts to express, then you can find that total SCC1 will be there, SMC1, SMC3, SCC1, PDS5, SCC3 and SCC2, SCC4 will bind to there. After that only this entire complex can be loaded into the chromosome. So just a minute, I think someone is writing there. Yeah. Yeah. So that's why cohesin can only be loaded in the late G1, because SCC1 will be produced in the late G1. So here first cohesin in early G1, this is the structure of the cohesin in early G1. After that in the late G1, SCC1 production will be there. So SCC1 will come, and along with the PDS5 and SCC3, now this is the entire cohesin complex. After this SCC2 and SCC4 will come. Here is SCC2, SCC4 will come, and it will bind here. Also there will be two molecules of ATP will be bound. After ATP binding, this SMC1, SMC3 head will be come to the so the after the ATP binding, the head will be locked, or these two head will come to close, right? So this will be the orientation after the ATP binding and SCC2, SCC4 binding. And after that, this complex can be docked to the chromosome, because it has everything. It has total cohesin complex, SCC4, everything is there. So now it can dock to the chromosome. But still, in this docking, it could not entrap the chromosome within its ring. So for entrapment, it needs the ATP hydrolysis. As it has already ATP bound to here, so now ATP hydrolysis will be occurred, because SMC1 has a functional ATP as head, SMC3 has a functional ATP as head. So they will hydrolyze these two molecules of ATP, and after this ATP hydrolysis, this hinge region will be opened. DNA will enter through this hinge region, and after that it will be entrapped by the cohesin ring complex. And this is the final confirmation after the chromatid entrapment. So now after this chromatid entrapment into the when it will entrap the chromosome within its ring, now it is important to maintain this cohesion, right? Because there is always a presence of a negative regulator, which is WPL1. So WPL1, what does it do? It opens the SMC3 SCC1 this interface, and this interface is the DNA exit gate. So if it will open this SMC3 SCC1 interface, which is a DNA exit gate, then the DNA will be exit from here and it will come here, and the cohesin will be deloaded from the chromosome, right? But if you maintain the cohesion, then this should not be the case. For that, there is a protein called ECO1, which is a acetyl transferase, which transfer the acetyl group to the SMC1 here. Sorry, SMC3 here. This is SMC3. So it will transfer the acetyl group to the SMC3, and due to this reason, after this modification of SMC3, WPL1 cannot open this SMC3 and SCC1 interface. so it will be locked even in the presence of WPL1. So that's how this negatively regulatory effect of WB1 is prevented by this eco1 acetylation. So presence of this eco1, the even there is WBL1 is present, but in the presence of the eco1, the cohesion will always be established. But when in the at the onset of the anaphase, this cohesion should be lost, right? After the replication, you can see that after the S phase, there is two copy of the sister chromatid is there. So for the segregation, as you know that this SCC1 should be cleaved to destabilize this cohesion, right? So for this, separase will cleave this SCC1 subunit, and now the DNA cannot be entrapped within the cohesin. DNA will come out to the DNA will come out from the cohesin by this DNA exit gate, which is the SMC3 SCC1 interface. So now after the cleavage of the SCC1, now DNA is free from the cohesin, and now the two of the sister chromatids are not cohesed, so now they can be separated. And after this event, now there is a deacetylase called HOS1. This can deacetylase the SMC3 here. It will remove the acetyl group of the SMC3, and it will make the cohesin complex ready for the use in the next cell cycle. Okay, so after so the life of the SCC1 is it will start synthesizing in late G1, and it will lead to the cohesin recruitment to the chromosome. And after the after the anaphase, when anaphase has been started, then SCC1 will be cleaved by the separase and it will be turned over. Okay, and also after that, the HOS1 will remove the acetyl group from the SMC3, so that this molecule can be ready for the next cell cycle, right? So that is the mechanism how the cohesion establishment is there, and how this establishment is actually regulated here. So that is depicted here. So so is it clear? Any doubt here? Please let me know. Any doubt? You got it? How cohesion loading and how the cohesion is established? Anyone has any doubt? Please let me know. Just so if you have any doubt, just tell me, because it is very important to know you. Just go through the just go through the slide once again, and just let me know if you have any doubt at any step. So should I move on from this slide? Should I? Okay, so should I go forward from this slide?
Yes, sir.
Okay, fine. So is it clear, na? This slide? Okay, fine. So now from cohesive, now we can just jump to the condensate. Okay. So there are in the higher eukaryote or the metazoas, the condensate three kind of condensate was is observed. Mainly there is condensate 1, which has SMC2, SMC4. There is a glycine called cap H, and also there is two heat treated motif containing protein cap D2 and cap G. And in condensate 2 there is SMC4, but there is cap H2 in place of glycine. In place of cap H glycine there is cap H2, and there is two heat treated and there is two heat treated motif containing protein cap D3 and cap G2. Okay. And in the budding yeast scenario, you can see that there is two in the condensate budding yeast condensate complex. But in the budding yeast condensate complex, there is two. The architecture is there is SMC2, SMC4. There is a glycine BRN1 which interact with SMC2 and SMC4, and BRN1 here in the higher eukaryote it is cap H, and in the lower eukaryote binding is it is BRN1, nothing else. And there are two other protein which has the heat repeat. They are DNA binding protein YCS4 and YCG1. That is the binding yeast condensate complex architecture, and this is the architecture of the condensate for the higher eukaryotes. So so recently in 2018 it has been found that condensate has the role of, and they did this single molecule imaging. By the single molecule imaging they showed that loop extrusion of the activity of the condensing. In this paper it was published, science paper. So the experiment what was carried out here, they have taken the quartz plate. Okay, in the quartz plate was incubated with streptavidin, and a DNA molecule was tethered by two ends to this quartz plate, and the two end of the DNA contain the biotin. So as you know that the biotin streptavidin, this makes a very strong interaction, covalent. Yeah. So this DNA was tethered to the quartz surface by this biotin streptavidin interaction. After after tethering the DNA in this surface, they found the loop extrusion activity by adding condensate and ATP. So after adding condensate, they found that there is a loop extrusion in the DNA. Here you can see there is a loop extrusion of the DNA, and also in addition to that they have also checked with the condensate whose ATPase head domain was mutated. So ATPase mutated head domain containing condensate failed to extrude this loop. So that's why from this experiment it was evident that ATP and condensate is required to extrude the loop here, and which is also known as the role of the condensate to make the chromosome more compact or structurally organize the chromosome. So now they tried to characterize the extruded loop by the condensate. How did they characterize? First they studied that whether the loop which is being extruded here, whether it is symmetric or asymmetric. So for that suppose this is your DNA. Okay, this is region 1, this is region 2, this is region 3, suppose. So the region 2 actually the loop formation is occurring by after addition of condensate and ATP. So this is the graph for the y-axis is the DNA size and x-axis is the time frame. Okay, so if you find yes, if you study for the DNA region 1, throughout the time region 1 DNA size is same. Okay, but the loop which is defined by the region 2, this region 2 DNA size is being increased initially and then it becomes saturated, right? So that means that initially from 0 to 30 almost second, in this time period the the region 2 DNA size has been increased. Region 2 is nothing but the loop, so the loop size has been increased initially. And other region we have region 3. This region 3, it has been decreased. That means what? That this region has been decreased, this region loop has been increased, and okay, so I mean the DNA size of this region is decreased, DNA size of this region is increased, and DNA size of this region remain constant. What does it mean? What do you think? What does it mean? From which portion it is so from which portion the loop is being extruded, from region 3 or region 1? Hint is region 3 DNA size is going down, region 1 DNA size is constant, and region 2 loop size is increasing. So in this loop formation, from which region it is taking the loop? From region 3 or region 1? Can anyone have any guess for that? Hello? Anyone is there?
Yeah, is it region 3?
Region 3. Okay, region 3. Why? Why region 3?
Because the DNA size is increasing.
Yes.
So I thought,
Yes, very good. Because the DNA size from this region is decreasing, so that means it is taking from this region and this region, and the loop 2 is being increased. And this increment of the DNA is taking from this region 3 because it is decreasing, right? But the region 1 is not decreased. That means it is only taking the loop from this region. Okay, very good. So that's why this loop extrusion was taken from a particular site, which is region 3 here, not region 1. That means this loop extrusion by the condensate for in this experiment is asymmetric. If it was symmetric, then we would have seen that by at the same rate the loop DNA has been taken from this region 3 and this region 1 with the same rate. But this is not the case here. So that's why the loop extrusion by the condensate is the asymmetric here. So any doubt here? If you have any doubt, please let me know, because it is very important to understand here. Any doubt from this experiment? Why it is asymmetric? Need to understand that why it is asymmetric. Okay, so I think you have understood. Okay, so let's move on to the next slide. Okay, so here again another experiment they did, where so till now they found that the loop extruded by the condensate is asymmetric. But now they have had a question that whether this loop extrusion is mediated by a single condensate molecule or multiple condensate molecules are involved in mediated by a in mediating this loop extrusion. So is there single molecule or there are multiple condensate molecule? So to answer this question, they designed one experiment. So what they did, they tagged the DNA by Ctex orange, which is a fluorescence dye, and which emits the green light, and they tag the condensate with a fluorophore which is ATTO6479, which emits the red light. So initially from a individual channel, they have this is the in this DNA channel, I mean the Cetox orange channel, you will find the DNA, and in the ATT0479 channel, you will find the condensate, right? Red, and this is DNA. So when you will merge this two channel, DNA and condensate, Cetox orange and this channel, so now you can see that there is this green one is DNA, red one is condensate which is unbound. But when there is DNA condensate bound or interaction, then you can find a merge of green and red color, and merge of green and red will give you yellow color. So that means here you can see a yellow spot here. That means that is the position of condensate which which sits at the neck of the loop here. Like here they have showed in the model, condensate sits at the neck of the loop, and it will extrude the loop like here. Okay. So by this experiment they found that condensate sit at the stem or the neck of the loop, and it will extrude the loop. Okay. So in this experiment they found the position of recruitment of the condensate. Now the remaining question was whether there is single molecule of condensate or multiple molecule of condensate is mediating the loop extrusion, right? So for that they use the concept that every fluorophore has a bleaching life, right? Every fluorophore has a lifetime, right? So after some time that fluorophore will be bleached. So they in different they did so they measured the intensity intensity of this condensate attached fluorophore. So they measured the intensity for this particular time period, and after measuring they found the graph. In the graph, initially from 0 to 25 seconds there was no condensate, so there was only the background channel. At the 25, when they added the condensate, they found the fluorescent signal, which is which corresponds the ATTO647 dye, or the ATTO647 fluorophore. That is after 25 to 50. Then after at the 50 second, they found that the fluorophore was found to be bleached, and there is again no signal, and it is the background signal. Okay. So in this event they saw that there is only one cycle of the the fluorescence and the photo bleaching. This cycle repeated only once. That means there is only one condensate molecule who actually mediating this loop extrusion. Because if there were more than one molecule, then this cycle was not coincident with I mean this cycle of the condensate this condensate attached fluorophores fluorescence and the photo bleaching. This cycle was not same for the each of the condensate molecule. If there were many condensate molecule, so if there were more than, or if there were other condensate molecules also, then we could have seen that one photo bleaching, and immediately after we could have seen that another or it was possible that for the first condensate molecule you find this cycle, but in the second condensate molecule you could have find that there is again a fluorescence activation, and then again a bleach. So this typical pattern or this typical cycle would be repeated if there would be more than one condensate molecule mediating this loop extrusion activity. But here only one, but here within this time period only one this kind of cycle of condensate fluorescence activation and then photo bleaching. So this cycle was repeated only once here. So that's why they concluded that there is only one condensin molecule, only one condensin molecule. It is in the molecular level. That's why it is single molecule imaging. And that they told that one condensin molecule in this experiment, what they did, which is mediating this loop extrusion activity. And if you will see the video here, you will find that before playing the video, there is they stain the DNA by Cetox orange. They attach the fluorophore to the condensin, which will emit the red color. DNA will emit the green color, and if there is any interaction between the condensin and DNA, that position will be yellow color, which is denoted by the arrow here. So now, if you will play the video, you will see that initially there is DNA, there is no trace of condensin. Now, if you can see that at this position, a loop is being extruded here, and the color is yellow, right? Sometimes the color becomes yellow. That means that is the junction of the condensin and the green and the red, which will make it yellow, right? So here condensin is being recruited at the neck of the loop, and yeah, and it will extruding the loop, which is evident from this video also. Just follow the loop region here, yellow color. That denotes the condensin recruitment. Okay. So can now anyone tell me what two important conclusion was made by this experiment here? What two major conclusion about the loop extrusion activity of the condensin was made here? Can anyone tell me? Two major points just.
So the yellow marks the meeting of condensin and DNA
Yes, very good.
because they were stained by two different color dyes.
Very good.
And the second point is that there's a single condensin molecule, as it is visible from the graph. There's only one high point. If there are multiple, there are multiple.
Yeah, there is one cycle, right? There is one cycle of this fluorescence activation and the photobleaching, right?
Yes.
Yeah, so that's why there is only one condensin molecule is involving here, and the position of the condensin, you can see that it is at the neck here, at the stem or the neck position of the loop here. If you will follow the position of the blue dot here, it is just at the neck here. So that is also a conclusion from this experiment. And from the previous slide, sorry, from the previous slide, what was the main conclusion from this experiment about the loop extrusion by the condensin?
Asymmetric.
Asymmetric.
The whole process is asymmetric.
Asymmetry loop extrusion, very good. And also here you can see that both condensin and ATP you will need to extrude the loop here, right? Yeah, nice, very good. So that was so that's it for today. So now just let me know if you have any doubt about any point of our discussion.
So, sir, I have a doubt, but it's not regarding week 6. It's a general doubt. So,
Yeah.
I want to know that I can ask.
Yeah.
So, my doubt is regarding chromosome organization.
Sorry?
So, it's regarding chromosome organization.
Chromosome organization. Okay.
Yeah. So, so we have a 23 pair of chromosomes.
Huh.
I want to know how they each separate pair, how they are organized. Are they organized as separate like pictures of replication, we only see a single like linear pair, but at the same time you have two different like sets of like chromosome. One is maternal and one is paternal.
Huh.
Like both of them replicate at the same time, or like are they like both of them like arranged like together like one whole linear set containing maternal then paternal or different entities? That was just confusing to me. I read about that, but I couldn't get a like answer that makes sense. Like maybe some like correct. You can correct me if I'm wrong. Like
Okay, so can you please take care of your mic because your voice sometimes broke down, so I was not able to listen to completely.
okay, so
Can you please repeat?
yeah, like my question was regarding chromosome organization.
Yes, yes.
Yeah, so there is a maternal and paternal chromosome,
Huh,
right?
huh,
We have a 23 pair.
huh, huh,
One is from maternal, one is from paternal.
huh.
So, like when we are talking about replication and all the slides and all the things, we see one of the DNA strands replicated.
Yeah.
Like and like at that time, both maternal and paternal chromosomes are replicated, right?
Yeah, in the S phase, both of the maternal and paternal chromosomes will be replicated. Yes.
So the whole process that we talk about, like both of them are replicated.
Both of them are replicated.
Yeah, and when talk about chromosome organization,
Huh.
like how are those two organized? Like are they different from each other, or is it like a big stretch of chromosome where the
Yeah.
first half is either okay?
Yeah, so you're talking about the diploid cases. So here in the diploid, one chromosome is from your paternal and one is maternal, right? So in the paternal, there are 23, for human scenario, there are 23 chromosomes, right? So, so there is common, yeah. So this 23 chromosome will make a complete paternal chromosome, and the structural organization within this paternal chromosome will be within this paternal chromosome. Okay, and for maternal chromosome there is also 23 numbers of chromosome in your uh, maternal chromosome, and this 23 will be organized separately from the paternal. So there is, yeah, so there will be internal chromosomal organization of the paternal chromosome, and there will be a separate internal uh, chromosomal organization in the maternal chromosome. So that's how you will get 23 plus 23 46 chromosomes.
Okay, okay. So both are replicated independent of each other, right?
Yeah, both are replicated. Yeah, but both of them replicate in the same phase, S phase. Okay, in the same phase, but the replication was, I mean, separate. They will be replicated separately, and their chromosomal organization will be also maintained separately.
Okay, okay.
Yeah. Okay. Any more doubt in any topic? Anyone else, or anyone if you want have?
So if you have no doubts, can we leave?
Yeah, yeah, yeah. You can leave. Session is
Thank you,
over.
sir.
Anyone else have any doubt? Let me know. Please. Okay, okay. Okay, so I think no one have any doubt for today. Okay, so let's close the session. Yes, you can leave, and so, see you on the next week.
Okay, sir.
Okay.